This is the acetylcholine signaling pathway by Sky and Manasvi. Our radio number is 104. And the acetylcholine signaling pathway is an important process in the nervous system responsible for transmitting signals between cells. Acetylcholine, a neurotransmitter, binds to specific receptors on target cells, including neurons, muscle cells, and other tissues, triggering various responses such as muscle contraction, heart rate regulation, and cognitive functions like learning and memory. In this video, we'll focus on the pathway's influence on muscle contractions. So first, some information about the pathway. The signaling molecule in the pathway is acetylcholine and is hydrophilic due to its polar groups. It's found in the central nervous system and the peripheral nervous system, and is responsible for communicating signals between the neurons in the nervous system. It enables cellular processes for muscle contractions, memory, cognition, and attention. So some other information about the pathway. So advantages of the pathway include that they enable fast and efficient signal transmission, allowing for reflexes and other rapid movement. They're also important in muscle memory, and the receptors can adapt to changes in neuromuscular plasticity. The disadvantages include that the acetylcholine receptor dysfunction can lead to weakness and fatigue in muscles, and there are many toxins and poisons that can lead to excessive activation of acetylcholine receptors, which is often deadly. And how it works is that acetylcholine signaling proteins are released by motor neurons that bind to acetylcholine receptors that are found on muscle cells. The binding opens ion channels on the cell membrane, allowing sodium ions to enter the cell and initiating a series of cellular reactions, which ultimately leads to muscle contractions.
We're starting with the signaling pathway components now. First of all, the cell's intention is to contract and release its muscles. So first we have the stimulus, and the stimulus is the action potential. Then we have the specific signal, and that is the acetylcholine, ACH, which is going to be released by motor neurons, and it's going to trigger the muscle contractions. Then we have the receptor, and the receptor is NACHRS, which is going to create a pathway for Na plus to enter, and this is going to set in motion the process. And there's also MACHRS, but we're going to focus on NACHRS in our model, and then we're going to form G protein coupled receptor complexes in the cell membrane. Then molecules that are also included in this pathway is sodium ions, Na+, which is going to cause depolarization, and then calcium ions, which is Ca2+, which is going to be released through our YR1, which we're going to get into later, and it's going to be released through SR, which we will also get into later. And then it's going to be released into cytoplasm. And then we have myosin, which is going to bind to actin, and that is what's going to drive the contraction. And we have ACHE, which is going to interfere with ACH, and this is what is going to cause the muscle to relax. This is the termination step. Now, before I show it to you, I'm going to explain how the pathway works. So we start off with ACH being released from the motor neuron, and it's going to bind to its site. And then once binded, Na plus is going to be able to enter the cell and start depolarization. And then DHPR is going to activate RYR1, which is going to release the Ca2+. And then once this is released, it's going to go into the cytoplasm. Then we have myosin, which is going to bind to actin, and this is what truly causes the contraction. And then ACHE is going to block the ACH signal, and that's what's going to cause the process of termination, which is going to cause the muscle to relax. When this happens, Ca2+ is going to move back into the SR, and then myosin is going to detach, and this is going to cause the muscle to relax. So we're inside a muscle, and this is the end of a nerve cell, and these are vesicles that are holding acetylcholine, these blue dots. Acetylcholine is going to be released, and it's going to come and it's going to adhere itself to these acetylcholine receptors. Once this receptor has been bound to it, it's going to be able to open itself. Now, Na plus is able to enter the cell through these channels. Also, the action potential is going to come, and it's going to come all the way down here to the T tubule, the T tubule, and in here, this orange receptor, which is DPHR, is going to detect the activation potential, and it's going to cause RYR1 to open, and now these yellow dots, which are Ca2+, can now come out into the cytoplasm. Once in the cytoplasm, they're going to connect themselves to this, and what this is this top here is actin and the bottom is myosin. That is why, when connected, it is called actomyosin, because of, as the name says, these two are combining. Once this happens, these bridges are going to form, and once these bridges form, contraction happens. Now, in termination, what happens differently is that when these acetylcholines try to come out, they can't enter the receptor because these pink items, which are ACHE, are going to prevent them from being able to bind to a receptor, and therefore the reaction cannot happen. When this happens, this means that the cell, the membrane is going to, these channel proteins are going to close, Na plus can no longer enter, and K plus is going to leave the cell. Also, the action energy is going to go back to normal, which means that DPHR is no longer going to be able to receive the signal that it's there. So RYR1 is also going to close, and now Ca2 plus can't come out anymore. So what happens? Ca2 plus, these yellow dots here, are going to go through against the gradient an ion channel into the SER, and the SR, and that is with the help of SERCA, which is going to pump them into the SR so that they can reset for the reaction. Now, without this Ca2+, the actomyosin isn't going to happen, so these bridges are not going to occur, and this is what is going to cause, so no more bridges, causes the relaxation of the muscle. So to recap termination, acetylcholine is going to be broken down by acetylcholine esterase, and the Na plus is going to stop polarizing, and the gated ion channels are going to be closed, and therefore K plus is going to exit the cell. Then Ca2 plus is going to stop being released, and instead it's going to go back into the SR with the help of SERCA, which is going to pump it back to the SR. And then the binding sites and the cross bridges that are being created by myosin and actin, those are going to stop. So the whole reaction is then terminated and ends.
Atracurium is a neuromuscular blocking agent often used in anesthesia. It's a skeletal muscle relaxant that's used to relax muscles during surgery or while on a breathing machine. It's a nicotinic antagonist, meaning that it blocks nicotinic receptor proteins on muscle cells. It's exogenous, as it's a drug that's not produced by the human body. Atracurium is an antagonist. It competes with acetylcholine neurotransmitters to bind to the acetylcholine receptor proteins. When it binds to the acetylcholine receptor proteins, it stops the ligand-gated ion channels from opening up. This means that the sodium ions cannot enter the cell. When the sodium ions do not enter the cell, there is no action potential that is formed. When there's no action potential, the DPHR in the T tubule will not detect the action potential, and it will not activate the RYR1. When the RYR1 is not activated, the calcium ions in the SR do not get released by the RYR1, so they stay inside the SR. And as there's no calcium ions in the cytoplasm, this means that the sarcomeres cannot contract and the actomyosin will not be activated. This means that the overall cell response will be muscle relaxation. Thank you for watching.